Another clinically useful tool to assess whether or not a given antibiotic is going to work in a specific infection is the Kirby-Bauer method. Those of you that took microbiology in undergraduate, excuse me, may remember this specific test. What you do is you take a sample and streak a few plates, and then once the bacteria grow and fill the plate, what you do is you add little discs, little paper discs that have been soaked in various concentrations of specific antibiotics, and you incubate that overnight. And you can see the more potent antibiotics have a larger what's called zone of inhibition around them. So theoretically, the drug diffuses out from the paper and kills or reduces the growth, but more often kills the bacteria excuse me, around that disc. And so what this is basically a really nice way to kind of assess whether or not a specific antibiotic is going to be more or less potent. So you can see, so this one, for example, here is as trianam. That is a very large zone of inhibition, and this is in contrast to something like ampicillin, which has a much smaller zone of inhibition. So in terms of bacteria cell killing, we'd go for something like astrianam for this specific patient. So this kind of feeds into a little bit of natural selection. Of course, this was first proposed by excuse me, Darwin, talking about basically that within every population, from bacteria to butterflies and everything else, there's a group within that population that is inherently resistant to a specific drug, to a pathogen, to some type of environmental insult. Okay, and so what happens here is that you have a large group of bacteria, and all of the blue ones are essentially normal bacteria, and then this orange one is the one that's resistant to a specific antibiotic. Once the antibiotic is treated, or is used to treat an infection, you can see that 80 to 90% of the regular bacteria succumb to the antibiotic, leaving one remaining normal bacterium and one remaining resistant bacterium. What happens is that over time, the resistant bacteria start to fill this environmental niche that's left behind in a much faster rate than the previous bacteria that were blue. And this is basically artificial selection working at its greatest. And so what you have is that the entire infection basically switches from a sensitive blue strain to a resistant orange strain. And so this is graphically, so this is pictorially shown here and then graphically depicted here. And you can kind of see that the total population of bacteria is this red dotted population. The sensitive population plummets shortly after an antibiotic is administered, while the resistant population continues to grow unchecked. And so this is kind of really how drug-resistant pathogens become problematic. This is also a justification for, again, the Goldie-Coleman hypothesis, which was discussed in a previous video, basically saying that in order to properly kill all these bugs, you should use one agent that is specific for the blue bacteria, excuse me, and another agent that is specific for the orange bacteria. So that's this way you'd kind of cover your bases. And so in terms of resistance mechanisms, there are some that are intrinsic, which is what I just showed you. There are some organisms that are just inherently resistant to any drug. There are also acquired resistant mechanisms. And so here you can see kind of mutations and genetic shift occurring at a much faster pace. And so with the acquired resistance mechanisms, we're looking at mutations. And so what the bacteria does is it changes the binding site of a specific molecular target that is known to bind to the drug. So we'll talk about where all these drugs bind to in the different bacteria. All bacteria do is just change the binding site so the drug can no longer bind to that target. Drugs can adapt. They can change the constituents of their cell membrane. They can change the chemistry of their cell membrane so that certain drugs cannot get in. Great example of this would be the production of the multiple drug resistant pump, also known as the ATP binding cassette or ABC pump, also known as P-glycoprotein. It's the same concept. It's got 3 different names depending on who discovered it and what discipline they come from. Gene transfer is basically the transfer of specific genes that govern resistance, more often by way of plasmid, or in some cases bacteria to bacteria. Basically that allows them to survive a specific insult from a given antibiotic. There is cross-resistance where there's enough chemical similarity between some of the antibiotics where resistance to one type of class confers resistance to all the other drugs within that class, and that kind of refers to the innate chemistry of these compounds as well as other chemistry, things like van der Waals forces, steric hindrance, and so forth. All things you remember, I'm sure, from organic chemistry. I want to talk briefly about some of these acquired resistance mechanisms, and it's really important to bring up P-glycoprotein. It's got different names, multiple drug resistant pump, ATP binding cassette, CD243, P-glycoprotein. It's the same concept. All of this is a protein that exists in the plasma membrane of either mammalian or eukaryotic or bacterial cells, any of them. And its job is to simply pump out foreign molecules. So foreign is relative, obviously, to whatever organism is producing it. But within that realm of foreign molecules are the antibiotics. And so if the antibiotic can get into the cell, P-glycoprotein essentially uses energy in the form of ATP to actively pump these chemicals back out. Okay, and so these protein pumps are found in the intestinal epithelium. They're found in hepatocytes, different portions of the kidney, as well as specific barriers within the endothelial cells, particularly the blood-brain barrier and the blood-testes barrier. These drugs are important in terms of cancer chemotherapy as well as antibiotic resistance components because they all confer resistance to a given drug, whether this be an antibiotic, an antiviral, or even an neoplastic agent for cancer. It's the same concept. These proteins have a high specificity to foreign molecules. Many foreign molecules have a unique fingerprint that identifies them as foreign, and P-glycoprotein actively finds these molecules and pumps them out. ATP binding cassettes in mammalian cells are more often called P-glycoprotein. The bacterial gene that is responsible for this is the bacterial LMRA. It's the same concept, and so depending on who you read and what their background is, whether it's molecular biology, immunology, microbiology, or pharmacology, the concept is the same, and it's really important. Other things that can reduce the activity of the antibiotic. So in the previous slide, we were talking about the chemical antibiotic itself being unchanged and just actively pumped out. Bacteria can produce specific enzymes that can pharmacologically or chemically change these drugs to render them inactive or at least reduce their activity. And a great example of that would be the enzyme beta-lactamase, which I'm sure you've probably heard of. So beta-lactam really refers to this ring structure within the penicillin molecule, and it's not really a ring, it's actually a square, but you get the idea. This is the beta-lactam ring. Okay, beta-lactamase is an enzyme that's made by bacteria to break this bond and reduce this bond, and you can see this compound being formed here, penicillic acid. This has a reduced activity in terms of bacterial cell killing, and so what this beta-lactamase enzyme does is it actually renders penicillin inactive. Okay, and so it deactivates it, and then it obviously, if it's inactive, it cannot kill bacteria. And so this is kind of a great diagram depicting kind of the rise of resistant bugs, and you can kind of follow this by yourself. I'm not going to go through this whole thing because this is essentially what I've been talking about, but basically what you see here is natural selection or artificial selection, selecting for a multiple drug resistant type of parasite, and then how it gets incorporated into the food chain, of course, and then how persistent and often inappropriate use of antibiotics kind of produces these drug-resistant superbugs. Great places to find these include within the food chain, so the farming industry, of course, as well as the hospitals and nursing homes, unfortunately. And so another kind of way of looking at it is how the epidemiology of some of these compounds get incorporated into the food chain. And you can see there's lots of things to think about. There's aquaculture, how these drugs can get released into the water system. You can see how they can get incorporated based on extracurricular activities that include, swimming and beach going. Companion animals are reservoirs of antimicrobial resistance, as are food animals. And then, of course, sewage and human waste removal, and how it interferes with, or how it's implemented in terms of the fertilizer industry. All of these are specific reservoirs of how antimicrobial resistance kind of persists in the food chain and then gets incorporated into, different types of care settings. So it's really kind of a problem. The bulk of the new antibiotics that are coming to market now are actually coming from medical centers like Rush and the University of Chicago and so forth that have active research programs that are looking for some of these new compounds. I'm affiliated with one of them, but I'm looking at them from a cancer perspective, of course. The bulk of the drug companies typically don't look for antibiotics simply because the payoff is low. There's a lot of obstacles, there's a lot of resistance. Usually by the time a new drug is released for or gets approved for human use, there's already resistant strains that have been identified. Okay, so we'll end here and we will continue on with this introductory lecture.